Okay, so today we will talk about gram staining. And this is the last topic to cover, right, under the heading of prokaryotic cells. So, gram staining is a technique that was developed by Christian Gram. Christian Gram. And since that's his name, right, when we write gram staining or use this term in a variety of ways, the G is capital, right, because it comes from a proper noun. And it is a technique to identify the types of bacteria. Well, one technique. And it involves their cell wall. And right, as a result of this technique, which I'll just get into, what you get is either you identify the bacterial cells as gram positive or gram negative. And that helps identify which antibiotics can be used against them, right? Right. So obviously, we're concerned about pathogenic bacteria. So which antibiotics would work? Because the result of the gram stain test tell us something about their cell wall, the structure of their cell wall, right? And that will become clearer when we talk about how this technique actually works. And when we know about their cell wall, right, we can work out, okay, fine. So which antibiotic do we use to treat the ailment and get rid of the pathogen? So this is how it goes, right? The technique. The bacterial cell walls are initially, they are stained with the gram stain. Right, and the gram stain is a complex of crystal violet and iodine. Right, the gram stain is a complex of crystal violet and iodine. Then you wash them with ethanol, right, the bacteria. So wash the bacterial sample cells with ethanol, and that ends up dehydrating. And then you apply another stain, which is the red safranin counter stain. And then you check results, whether they are gram positive or gram negative. Now, which one they are, right, depends on the structure of the cell wall. So a bit of suspense before I tell you which one's which. All right, so the bacterial cell wall, right? Not as an organelle, but in the context of the gram stain. So the bacterial cell wall can be of two types, right? It can be of the type where the cell membrane, right, or the plasma membrane has an outer thick coat of peptidoglycan. Right, so the plasma membrane has a thick peptidoglycan layer on top, right? And within that are chemical substances such as teichoic acid, right? So containing teichoic acid. That is one type of structure that we could possibly come across. The other is where, right, there are two parts to the membrane. So there's an inner membrane, right, for the plasma membrane, right? Then there is a thin peptidoglycan layer, and then there's an outer membrane. Now, what happens is, right, first you put in the gram stain. Right, which is again the crystal violet. An iodine complex. And this complex is absorbed by whatever layer is on the outside. Absorbed by outer layer. And I'm saying layer, not membrane, right? So it could be the thick peptidoglycan layer, it could be the outer membrane, right? So it is absorbed by the outer layer. Then when the cells are washed with ethanol, right, two things can happen. Right? If there's a thick peptidoglycan layer, right, it's not dissolved, it's not washed off. It's not washed away by the ethanol. And in that case, right, right, so the other option is that if there's an outer membrane, right, outermost layer is the outer membrane, right, it is washed away by the ethanol. So there are two things that can happen depending on how the cell wall is. Then comes the third part, right, which is the red safranin counterstain. So in case of, right, the first scenario where the peptidoglycan layer is not washed off, right, the cells do not absorb this new counterstain. and so under the microscope they appear blue. Or if the outer membrane was washed off by ethanol, right, then the stain, the counter stain, is then absorbed by the thin peptidoglycan layer that comes next. Right, and because it's red saffron, under the microscope, these bacterial cells will appear to be red. Why? Because, right, in the first case, the blue dye, the crystal violet iodine complex, the gram stain, was absorbed by the peptidoglycan layer, right, and it wasn't washed away, so they were, right, blue to begin with, and they were not affected by the counter stain, and so they appeared blue. In the second case, right, the gram stain was absorbed by the outer layer, the outer membrane, sorry, right, and then it was washed away by ethanol, so whatever color the cells had gotten because the staining is gone, because the cells are naturally colorless. Right, and so now they've lost their color, and when treated with the red saffron and counter stain, that is absorbed by the thin peptidoglycan layer that came after the outer membrane, and so they have a new color and they appear red. Now, which one's which? Well, the blue ones are gram-positive bacteria. Right, so these guys are gram-positive bacteria. And these ones that appear red are called gram-negative. Clear?
Yes, sir.
Right, so that is gram staining. Now, the thing is that just by using the gram stain, it is one way of figuring out the type of bacteria, but it's not entirely useful in other ways. And so there are other categorizations of bacteria, right? So other ways of identifying or characterizing bacteria. So the bacteria can be classified according to their shape. And there is a variety. Right, so some bacteria are spherical. So those are called foci. Then some are rod shaped. And so those are called bacilli. And these are all plural, right? So foci is actually focus for singular, right? Bacillus for singular. And then they could be twisted, in which case they're called spirilla. Or they could be comma shaped, in which case they're called Vibrio. Right, and so you might come across names such as lactobacillus, right, and this is in the production of yogurt, right, so lactobacillus and staphylococcus. And so this is actually telling you the shape, right? So these guys are rod shaped, these guys are spherical, and then, right, Vibrio cholera. And this is the bacteria that's responsible for causing cholera, right, which can be fatal, right? And there's so, right, some of these bacteria, right, because the classification doesn't depend on them being pathogenic and which antibiotic that would be useful against them, but depends on their shape. And so, right, some of these bacteria are actually used industrially for different processes. Some are obviously pathogenic, right? And so, when you read the names of bacteria, and right, if you want to get a taste of that, you can take if you've got like any antibiotic at home, and you have that leaflet that gives you all the details. You can take that out and you can start reading it, and they mention something about gram positive and gram negative, and maybe you come across some of these words, right? The focus or the bacillus or something, and that would start making much more sense to you, right? So you know a bit about the kind of bacteria you're dealing with. Another way of grouping these guys, right, is talking about their oxygen requirement. So there are three kinds, right? There are the kinds that are called obligate aerobes. And aerobe means oxygen, and obligate means necessary. So these bacteria need oxygen for respiration, right? They can't do without it. Then you have the facultative anaerobes. So these guys use oxygen if available, right? But can function normally without. Right, so they can do well in anaerobic conditions, and right, the book mentions that most pathogenic bacteria fall into this category. Right, most as in not being the most dangerous, but the quantity of pathogenic bacteria classified as pathogenic, right, are actually facultative anaerobes, right? By number. And then the third is the category of the obligate anaerobes. And so these guys are crazy, right? They only function without oxygen, right, in anaerobic conditions. In fact, oxygen will actually kill them. Oxygen is toxic to them, right? And so they have to, they can only respire. The respiration only works in case of anaerobic conditions. Is all of that clear?
Yes.
Okay, good. Now we move on to last chapter of section 3A, 3A5, which is the organization of cells. Right, and the information given here is pretty basic. It's right, almost like what you have in your IGCSE or O level. And they mention that multicellular organisms have obviously specialized cells. Right, so specialized cells are the ones that develop during development. They develop or differentiate, as the word is, to perform specific functions. And when a group of these come together, so a group of specialized cells forms a tissue. And then many different tissues come together to form organs. Right, and you've got when you've got many different tissues coming together, that means you've got many different types of specialized cells that are coming together to form organs. So this is the layer of complexity that builds from cells to tissues to organs, and then later on to organ systems. So a bit of detail about tissues. Right, we've talked about cells. Right, obviously we haven't talked about all the different kinds of specialized cells. Right, but those will come, some of them. So there are 4 basic kinds of tissue in the body, and those are the epithelial tissue, the muscle tissue, the connective tissue, and the nervous tissue. That is right by down. So there are 4 main types. Those are epithelial tissues, connective, muscle, and nerves. Okay, now these are the main types. So there are other types of tissues, but those are subtypes. Okay, so somewhere, if you move up, and so you can probably fit, for example, you've got cardiac tissue. Right, and so cardiac tissue can probably fit in here. It's a special type of muscle. Right, so you got lots of the subcategories, but these are the 4 main ones. And then there are, because the subcategories, there are modified versions of each of these. each of these. And so in the book, they give the example of different varieties of the epithelial tissue. Right, and as I refer to the book, that obviously means in your syllabus. Right, for the Edexcel. So the epithelial tissue are the ones that line the inner and outer surfaces of the body. Right? So these line the inner outer surfaces of the body. And right, a great example is your skin. Right, the outer layer of your skin is right epithelial tissue. Right, and it's actually the kind that is found compound epithelium. Right, so epithelial tissue. You also have squamous epithelium. And these are the ones that line the surface of blood vessels, right? Blood vessels. Then you have cuboidal and columnar epithelial cells. And right, you would find these on the surfaces of tubes of the gas exchange system. Basically, your trachea and bronchi. And also the oviducts. Then you have ciliated epithelia. And so these guys have hair-like projections. And often contain what are called goblet cells in between them. Right, and these goblet cells, what they do is they produce mucus. And right, the cilia move the mucus away from the lungs. Right. So those are the different kinds of epithelial cells, right? And then I gave you an example of connective tissue, cardiac tissue, connective tissue, cartilage tissue, and all of these, right, they can be taken to be as a sub type of the four main types. Then you've got organs, right? So as I mentioned, different tissues come together, they group together in a structure to form an organ. And right, so depending on right the type of tissues that come together, so right, not only the main types but the subtypes of those, right, the types of tissues determine what the function of the organ could be. Function of the organ. And then finally you've got organ systems. And this is when, right, multiple organs work together, right, for a specific, right, function, large scale, to perform large scale functions. And so obviously, right, this could include the digestive system, or right, the nervous system, or the hormonal system, right, or any other of those systems, right, the circulatory system. So any of these, and right, obviously, one organ can possibly be part of multiple systems. And it's not just humans that have organs, right? Plants also have organs. So the leaf is actually an organ, right? So plants also have organs and tissues. Tissues and organs, right? The leaf is an organ having different tissues. So they have, right, the vascular tissue, so the xylem and phloem. They have the epithelial tissue and the mesophyll tissue. Right, so recall to your IGCSE or O level. That's why not xylem tissue. Vascular tissue. And right, the epithelial tissue. And the mesophyll tissue. So that would include the spongy and the columnar mesophyll cells. And we're done with section 3A. So, right, or chapter 3A, right, as the book calls it. And now we're going to, right, not continue chapter 3B because, right, that's something we'll do later. It's not, we're not leaving it out, but we're leaving it for later.
Okay.
Right, so in our next classes for AS Biology, we'll be moving to chapter 1A from the textbook. And we'll start off by talking about, right, the importance, like the chemistry for biology, so what biochemistry is. And then we'll talk about biological molecules, where we'll discuss carbohydrates and proteins and lipids. All right.
Okay.
All right. So I hope that's all well understood. Do refer to your text, do review, and I'll see you next
Yes.
time, inshallah.
Inshallah. Allah Hafiz.
Allah.